Welcome to the fascinating world of computational biology. Today, we are going to explore nucleic acid databases. Imagine trying to build a massive, global public library, but instead of books, this library stores the instruction manuals for every living organism on Earth. These manuals are written in the language of DNA and RNA. We will learn what these databases are, how they grow, and the mathematics behind searching through them. To begin our journey, let us understand why these databases were created. In the early days of biology, scientists kept genetic sequences in their own private notebooks. This made sharing discoveries incredibly slow and frustrating. To solve this problem, scientists created nucleic acid databases. These act as centralized digital libraries where researchers from all over the world can upload, share, and search genetic information instantly. They store not just the nucleotide sequences, which are made of adenine, thymine, cytosine, and guanine, but also the associated biological metadata that gives context to these sequences. Now that we know what these databases are, let us look at the most prominent ones. The absolute foundation of this global sharing is the International Nucleotide Sequence Database Collaboration, often abbreviated as INSDC. This massive collaboration consists of three major databases located across the globe. We have GenBank in the United States, EMBL in Europe, and DDBJ in Japan. When a scientist discovers a new gene, they submit the sequence to one of these databases, along with annotations describing its function and origin. These three databases work together to ensure researchers everywhere have access to the exact same information. Building on this collaboration, let us examine how these three databases actually interact. On the screen, you can see a schematic diagram of the INSDC data synchronization cycle. Notice the three main nodes: GenBank in the USA, EMBL in Europe, and DDBJ in Japan. The thick double-headed arrows connecting them represent the daily data synchronization. This means that if a scientist, shown on the left, uploads a new DNA sequence to GenBank today, that exact same sequence will be copied to EMBL and DDBJ by tomorrow. This continuous cycle guarantees that the global web of biological knowledge remains unified and up to date. Seeing how seamlessly this data is shared brings us to why this infrastructure is so critical. These databases are the absolute foundation of modern biological and medical research. Consider what happens when a new virus emerges, such as the one that caused the COVID-19 pandemic. Scientists immediately sequence its genome and upload it to GenBank. This allows researchers globally to instantly download the sequence, understand how the virus works, and begin designing vaccines without having to sequence the virus themselves. Without this system, every laboratory would work in isolation, drastically slowing down life-saving medical breakthroughs. Additionally, specialized databases like the NDB store three-dimensional structural information, adding another layer of vital data. Because of this massive global participation we just discussed, the amount of data in these repositories does not just grow steadily, it grows explosively. Scientists are constantly sequencing new organisms. To manage this, computational biologists must model the accumulation of genetic sequences using the mathematical concept of continuous exponential growth. Understanding this explosive growth is absolutely crucial. It dictates exactly how much server space, memory, and processing power we will need in the future. If we cannot predict the size of the database, we cannot build the data centers required to store this invaluable information. To predict this future infrastructure need, We rely on a specific mathematical model. This is the formula for continuous exponential growth. Let us break down what each symbol means. N of t represents the total number of sequences in the database at a specific future time, t, which is measured in years. N0 stands for the initial number of sequences, present right now. The constant e is the base of the natural logarithm, approximately 2.718, which naturally appears in continuous growth processes. Finally, r is the continuous growth rate. If the growth rate r increases even slightly, the total number of sequences, n of t, will skyrocket much faster, demanding significantly larger server farms. Now that we have our formula, let us apply it to a practical scenario. Suppose GenBank currently holds exactly 200 million sequences, and historical trends show a continuous growth rate of 15% per year. We want to find the database size after exactly 4 years. First, we identify our variables and convert the 15% rate to the decimal 0.15. We substitute these into our equation. Next, we multiply the rate 0.15 by the 4 years to get 0.60 in the exponent. Using a scientific calculator, e raised to the power of 0.60 gives a growth multiplier of approximately 1.822. Multiplying this by our initial 200 million gives a final answer of 364.4 million sequences. This means the database will nearly double in just 4 years. With the database growing so rapidly, searching through it introduces a new challenge. When you search a massive database for a short DNA sequence, you might find a match simply because the database is so huge, not because the organisms are actually related. Since DNA is made of just 4 nucleotides, adenine, thymine, cytosine, and guanine, we can calculate the likelihood of a specific sequence occurring randomly. This mathematical check is critical. It helps us determine if a match we find in GenBank is biologically meaningful, meaning it shares an evolutionary ancestor, or if it is just a statistical coincidence. To perform this mathematical check, we use a straightforward probability formula. Here, the variable p represents the probability of a specific DNA sequence occurring purely by random chance. The number 4 in the denominator represents the 4 possible nucleotide bases in DNA, which are the building blocks of the genetic code. The variable L represents the total length of the DNA sequence we are searching for, measured in base pairs. Conceptually, this formula assumes each position in the sequence is independent. As the sequence length L increases, the probability P decreases exponentially. This means it becomes incredibly rare to find long matching sequences by pure chance. Let us see this formula in action with a concrete example. Imagine you are a researcher who has just sequenced a short, unknown DNA fragment that is exactly 10 base pairs long. You want to know the probability of this exact sequence appearing randomly. We take our sequence length, L equals 10, and substitute it into the exponent of our formula. This represents multiplying the 1/4 probability for each of the 10 independent positions. Calculating 4 raised to the power of 10 gives us exactly 1,048,576. Converting this fraction to a decimal gives a final probability of approximately 0.0000095. This incredibly small number tells us that finding this exact sequence by random chance is extremely unlikely. Now that we have calculated this tiny probability, let us interpret what it actually means for a biologist. A probability of 0.000000095 means there is only a 1 in over a million chance of this specific 10-base sequence occurring randomly in a genome. Therefore, if we search GenBank and do find a perfect match for this sequence, it is almost certainly not a coincidence. It strongly suggests that the sequence has been conserved by evolution because it serves a vital biological function in the organism. This is how mathematics directly validates biological discoveries. We have covered a lot of ground today, so let us review our key takeaways. First, nucleic acid databases are the essential digital infrastructure of modern biology, transforming isolated genetic discoveries into a globally accessible web of knowledge. Second, we learned that these databases grow explosively, and we can predict their future size using continuous exponential growth models. Finally, we saw how statistical probability helps us distinguish between random statistical coincidences and truly meaningful evolutionary relationships. By understanding both the biological data and the mathematical principles governing it, you are now equipped to appreciate how these massive systems drive modern scientific discovery.